I shouldn't have told you. All right. Multiple alleles. So all the traits we talked about before now were controlled by, you basically only had two choices. There was big A and little a. There are cases though, and I'll show you some more examples of this next week when we practice problems, where there's more than two possible things that you could give. Now, that doesn't mean that a particular person would be carrying more than two copies. You're still only carrying two copies. But the difference is there's more than two possibilities. So blood types, there are three different alleles, three different forms of the gene for blood type. There is I with a capital A, which codes for blood type A. There's I with a capital B, which is the code for blood type B. They are codominant. So if you carry one of each, your blood type is AB. And then there's little I, which basically codes for nothing. Little I codes for making no proteins. And if you're little I, your blood type ends up being O. Now notice how I wrote this here. An individual person is still only going to be carrying two copies of the gene. What multiple alleles means, it just means that there's more than two options of what they might be carrying. So IA, or IA little I is blood type A. That person would not know whether they were homozygous or heterozygous unless, like in my case, I know my dad was O and I'm A. So I know I would have to be IA little I. Since my dad was O, he would have had to have given me a little I. But other than that, if your blood type is A, you wouldn't necessarily know whether you were homozygous or heterozygous. Then B, then AB, and then there's only that one genotype for O. There is no benefit as far as like immunity or anything like that to what blood type you have. It makes absolutely no difference. No blood type is healthier than another. The only difference really is in the case of transfusions. If you needed a blood transfusion, then O would be the worst thing to be because you could only receive from other people who were O. That's why, because O, since it doesn't have any proteins, doesn't cause any bad reactions, so they use O in trauma. That's why blood banks are always begging people with type O to donate. It's not because O is rare. O is actually like 38% of the population. But if they bring you in on a helicopter in trauma, losing blood, they're not going to have time to figure out what blood type you are. But if they give you O, they know that's safe. So that's why they always want to keep lots of O in stock, because for trauma situations, they can give that to anybody. If you want the best blood type so that you could receive blood if you needed a transfusion, then AB. And then there's another gene, because everybody always asks me this, what's the whole positive, negative thing? Completely separate gene unrelated to this that codes for a different protein called the Rh factor. If you happen to be Rh positive, then your blood type is whatever they say, and then positive after. It means you have that other gene. And if you're recessive for the other gene, you're negative. So my blood type is A negative. So I know that I'm IA little I, and then I'm negative for Rh, so, negative, negative. You need to know how to do a cross with blood type. We're actually going to do two practice problems right now, so this should clarify it for anybody who needs help with practicing it. Wait, let me get your question first. I am not supposed to receive positive. As a matter of fact, if you are Rh negative, and you're pregnant, which I'm not, but when I was, if the baby is positive, they give you a shot of something called Rogam, because if you're negative and the baby is positive, apparently the deal is you won't have a reaction to the baby the first pregnancy. But if you get pregnant a second time, you can actually reject the fetus and have a miscarriage if the baby's positive and you're negative. So what they do is they give you a shot of this stuff, it's called Rogam. It's basically a shot that tricks your body into thinking that you've already made antibodies against the Rh factor. That way your body won't really make them, because if your body really makes them, they last like years and years. But the shot only lasts a couple of months. So they give you the shot, it tricks your body into thinking you made antibodies, then you won't make any, and then you're safe. But no, a person who's negative is not supposed to receive positive. My understanding is it's not as severe of a reaction. Like if I'm A and somebody gave type B, it would kill me, probably immediately. They gave me the wrong blood type, which would be a good lawsuit for someone who was left behind, but not for me. But if I'm negative and somebody gave me positive, my understanding is that the first exposure, you might not react at all. So it's possible that you could receive, and I'm not exactly sure why. I don't know if you just don't freely have those antibodies and you have to build them up or what. So, but anyway, that's depressing conversation. This is just a picture of the blood types. So here's two practice problems. Let's do the first one together. Let me Question? Because there's three, in every other trait, you basically could only get either big A or little a. In multiple alleles, there's more than two options. There's IA, there's IB, and there's little I. So there's three different options instead of just two. You personally are still only going to be carrying two letters, but there's more than two letters to choose from. Yeah, there are other traits that have even more. I know there's one for fur. It's called a chinchilla allele that codes for fur in, well, probably chinchillas also, but I think it's rabbits. And I think there's 4 versions of it. We'll practice with that too so you can see. All right, let's do the first one together. A male is heterozygous for type A. He marries a female who is type O. What kind of blood types can their kids have? So if he is type A, I know he's got IA. But he's heterozygous, so what must the other letter be? Little I. That's the only way he can be heterozygous and show type A. What would be the phenotype or the genotype for the woman who's type O? Little I, little I. Now you do the same cross. Nothing's new. This is really just exactly what we did last week. IA, little I, cross with little I, or you could do 4 boxes. What blood types can their kids have? That's it. So they can either be, there's a 50% chance of them being type A and a 50% chance of them being type O. That's it. So it's just a regular, it's just like a regular just like a regular cross. The way they write it is a little different. So try this one. A male who's type A marries a female who's type B. They give you a really big clue. This is massively important that their first child is type O. That is necessary to figure out the genotypes of the 2 parents. Exactly. And now they want to know what other blood types are possible in their kids. So take 5 minutes. All right. So the fact that they have a type O child, you can't solve the problem without knowing that information. The reason they tell you that is so you would know that the guy, even though he's type A, must be IA little I. He must be heterozygous. And the wife must also be heterozygous. Otherwise, there's no way they could have had a child who was little I, little I. So that's the reason they tell you the child. A lot of these, as I've said before, are kind of like logic problems. They give you little pieces of information. They want to see if you can put it together. All right, so if we do a Punnett square of this, IA little I, IB little I, usually in blood type crosses, they don't ask you for the genotypes and phenotypes. They just ask you what kind of blood types the kids can have. So what would be the blood type of the kid in the first box? So AB, what would be this one on the right? B, down here, A, and here. So they could have 4 kids and 4 different blood types in their children, which is kind of interesting. And I've actually seen that before, where I've had siblings that have different blood types, and 3 different siblings, 3 different blood types. So, and it's obviously it's not always possible. You can look it up online. I think A is like 34% of the population, and O is like 38% of the population. Those are like the most common blood types. B, I believe the rarest is AB. AB positive might be. That's only like 1% of the population or something like that. So again, it really doesn't do anything. It's just another factor on the blood.
And then you show that it's positive by not.
So, the positive and negative is not even included in this. So don't even worry about that. We won't even be dealing with that. All right, good and bad traits. So a lot of times we make the assumption that traits are bad, because they cause bad stuff to happen to you. So to me, that would make that a bad trait. But actually what we're going to learn about, and this exact example is going to come back in a couple of chapters when we get to evolution. Sickle cell is a really good example of where a trait that seems like it would be a negative trait, that it would eventually just disappear and get wiped out because it's bad, doesn't get wiped out. So people that have sickle cell, full-fledged sickle cell, it shows a little bit of incomplete dominance kind of thing. Because if you are a carrier of sickle cell, if you have 1 copy of the gene, from what I understand, you do have some mild symptoms. But if you have full-fledged sickle cell, little b, there's a lot of health issues. Your blood cells are shaped wrong. You're tired all the time. They're actually shaped. They crunch up in such a way that they're pointy and they can actually clog up your blood vessels. And the bottom line is that with no medical treatment at all, if you lived in some remote village in Africa with no medical treatment and you had sickle cell, it would be very likely you would die young. So we would make the assumption that if the gene is so bad, it shouldn't be very common because people that have it don't survive very long. And the bottom line is, that's not the case. If we look at Africa, 60% of people are carriers of the sickle cell gene. And what we discovered was, it is bad to have sickle cell anemia, but people who were heterozygous, which have very few health problems from it, are actually resistant to malaria. Now, malaria is not genetic, it's carried by mosquitoes, but malaria also is fatal without medical treatment. So it turns out that by having a copy of the gene, it actually gave them a survival advantage. It's actually called like heterozygote advantage or heterozygote superiority. There's actually a name for it that we'll talk about in evolution, but this is an example of where sometimes it seems like a gene might have a negative effect. There might be a reason why that gene is kept in the population. There might be something good about it that we don't even realize. And this is kind of neat, and then I'll answer your question. If you look at this picture, this is the areas of Africa where malaria is the most common. And if you match it up, notice how sickle cell is also very common in those areas. And all these green areas outside of that range, almost nobody carries the sickle cell gene. So carrying a copy of the gene actually gave them a survival advantage, and this is where we talk about natural selection and evolution and how genetics and evolution are very, very closely tied. And so sometimes a gene that might seem like bad might have a benefit to it, or a gene that seems like it has no effect at all, that it's neutral, might have a benefit to it we don't even realize. Did you have a question? Yeah, I'd have to wonder, because you would think he would know before he was that old if he was homozygous. That's a good question. So, last couple of things. Environment. Just a reminder, I said this at the beginning of the lecture. The environment plays a role in the expression of genes. In some cases, like with this rabbit, his fur grows in black only where it's the coldest. So, I don't know what scientist did this. Let's drop this ice pack here. But the fur basically grows in black because it's cold. So in theory, you could like shave goat canes into the side of your Siamese cat, and then, yeah, it's not very nice, but yeah. Now, somebody thought, just to clarify, his fur doesn't magically turn black. Like, you can't put him in the freezer for 5 minutes and take him out and he's black. It's that the fur grows in black when it's exposed to cold temperatures for a long time. I just want to clarify, because somebody in my other class thought the fur literally changed color when you put your hand, like a mood ring or something. It's not like that. And even more interesting, I think, is this, epigenetics. This is something we're just learning about now. So these two mice are genetically identical. The only difference is that their moms were exposed, they have two different moms. They were exposed to a chemical called BPA, but they have one mom had a diet that was supplemented with folic acid and B12 and some other things. The mouse on the right is completely healthy. The fur is obviously a different color. The one on the left has obesity, high cholesterol, a whole bunch of problems. Clearly. Genetically the same. In other words, what we're finding out now is that the stuff that unfortunately your parents did turns sections of your DNA on and off. So you might have the same gene code as somebody else, but your DNA is activated or deactivated during development by the diet or environmental exposures of your parents. And this is called epigenetics. And it's a whole area that they're studying now because it's totally beyond anything Mendel ever understood.
So is that why, like, obese parents might have obese kids?
That's part of it, but that could also be regular Mendel genetics, that you have more fat cells. Yes. Yes. No, this is like when you're in the womb, which is one of the reasons, like, for example, they're always talking about now folic acid being so important for pregnant women, because apparently the DNA activates to make your spinal column and your neural tube form, and apparently a lack of folic acid prevents that DNA from getting activated at the right time, so you can end up with like spina bifida or neural tube defects, not because your DNA was bad, but because your DNA didn't get turned on or off at the right moment during development. So, I don't know what foods, I know orange juice is high in it, but I know pregnant women, they usually have them literally take folic acid supplements these days. So, alright, that's it. The last of this is all practice problems, which I would like for you guys to work on for the rest of the period, and we're going to actually start going over them on Monday. If you want to skip to that one I was talking about with the parakeets, it's the last problem. If you want to see one that's kind of a harder problem, although this first one's kind of tricky too.